Hi everyone. Thank you so much for taking the time to hear me talk about our unique solution. And I hope by the end of this webinar, you will have some new ideas on how you can incorporate these unique solutions to your research. So just a quick summary of what I will be talking about. I will do a quick introduction about who we are and a little bit about our core technology, and then I will go into 4 of our unique solutions. And then at the end, hopefully, I can convince you to try out one of these products. So scRNA is a global company, as you can see here, and we were established back in 2018. And we have not only offering kits, but we also have services. So we offer services where you can send your sample to us. And as you can see, we have many locations across the world that you can do that. We're headquartered in Cologne, Germany. And we also have 2 sites in the US, 2 in China, 1 in Singapore. And very recently this year, we just set up our Cambridge site. So this is our core technology, the SCOPE chip, and it's a microwell microfluidic system. So all of our single-cell products are based on this core technology. As you can see here on the left-hand side, it's the size of a microscope slide. And we offer 2 types of microchip. Really depends on how many cells you would like to capture. But that's not all we do. We do offer what we call the end-to-end single-cell workflow, because we realize, sample preparation is just as important as the technology itself. So you could have, the best single-cell technology, but if your sample are not prepared properly, it doesn't matter which technology you use, the data you get will not be very good. And that's why we put a lot of investment and R&D into helping you get the best samples before you do the single cell part of the workflow. So we offer things from tissue preservation, tissue dissociation, to get to your single cell suspension, and then of course the cell partitioning library preparation. And we also offer bioinformatics support. So if you don't have in-house bioinformatic help, we can help you with that. But for today, I will only focus on this part, so the cell partitioning, the lysis barcoding, and to get you to sequencing ready library. So how does the SCOPE chip work? As you can see here, it's an open system, and all you need is a pipette. So you don't need an upfront investment on a machine. So it's a manual workflow, very easy, very accessible. So if it's your first time doing single cell, this is a really good way to get started. But it's also very good for people who are very experienced. And within this area here, you can see you have the microwells where you then help partitioning the cells. It's only using gravity, so it's very gentle compared to other technologies. So if you have very fragile cells, this is probably a better system for you. And the way we extract your mRNA is using a magnetic bead that contains your cell barcoding and your UMI. Of course, like many standard single-cell platform technology, we use the poly-A capture method, as you can see here. So how do you QC our microchip? It's actually, you don't need any specialist equipment. All you need is a light microscope. And as you can see here, this is what it looks like under a microscope with your NT wells. And then this is when it's populated with your cells. And then here's with the beads. And you do all the lysis and mRNA capture within the chip. And then a magnet is put on top of the chip to retrieve your captured mRNA. And as I mentioned, it's an open system, so you can QC at many different steps and makes it very flexible. So that's our core technology, but we go beyond that. So we offer things that are very unique multi-omics solutions, and here's a menu of everything that we offer. As you can see, there are a range of product we offer. But in the interest of time, I just wanted to focus on 4 of our very unique solutions that you probably cannot find anywhere else on the market. So I will start with our DynaSCOPE, which is our time-resolved transcriptomic at single-cell level. This is the only commercially available kit that you can find on the market, and it allows you to add the time dimension to your single-cell data. And how does it do that? It uses the S4U labeling technology. So some of you might be familiar, especially if you worked in developmental biology with scRNA-seq. It's a very similar way of doing it, but it's at single cell level. And all you need is one kit. So you don't need to try and play around with it and try to optimize it. It's all optimized for you. And we offer it for in vivo and in vitro study. So how does it work? With this workflow, you do the labeling before. So whether it's in vivo into your organism or in vitro in your cell line. And essentially, it converts the S4U tag into a cytosine analog. So any of your newly synthesized nascent RNA will be labeled with this. And this is how you differentiate between your nascent RNA and your older RNA. And that's what gives you that time dimension. So how does it perform? As you can see, with the kit, you will get your whole transcriptome data as well. So you will get that data, and then on top of it, the time dimension, all coming from the same cell. So you don't need to do separate workflow. And then you can then track your RNA metabolism and then differentiate your newly synthesized RNA and your older RNA. And as I mentioned before, it's really popular with developmental biologists. So if there's any developmental biologists amongst you, this might be of interest to you. But it also really good if you have a different condition, you want to track how it affects transcription. The next product is MobiuSCOPE, and this allows you to get full-length transcriptomic data. And what's different about this technology is that it uses short read sequencing. So you don't need to use long read sequencing, which is usually very costly. So this is a way to bring down the cost without losing the full-length information you need, such as splicing, variations, SNPs, fusion gene, etc. It also works very well with non-model organisms as well. So how does it work? So at the start, you still do your poly-A capture to get your mRNA. But after RT and PCR, you have, we'll split the library. So you will do your 3' library as you would as a normal step. But for the 5' library, we do a circularization step. This is a proprietary technology only SNVs has, and essentially brings your 5' end close to your UMI and your barcoding. And the reason for this is because we want to be able to give you data that is giving you true transcript data. What that means is when we do the fragmentation, and then we do the sequencing, and then when you do your bioinformatics, you know that these fragments from the 5' and the 3' are coming from the same cell and the same transcript because they have the same cell barcoding and the same UMI. And this is a way to give you true transcript data rather than just using random priming, random oligo DT, which means you don't actually know they're coming from the same transcript. With this technology, you can be confident that it is coming from the same transcript. So how does it perform? As you can see here, the coverage of the gene body is very good, and which you can get information that you would miss if you were just doing a 5' library or a 3' library and then try to reconstruct that. And here are just some data showing the advantage, especially in splicing junction, you do get more information. And then also with fusion gene, you do get better readout of your fusion gene than if you were to do them separately. And finally, I'm sure a lot of you, when I was going through that, will think, well, how long can you read through that RNA? Well, if you have really long RNA, would you miss that middle bit? Well, here, hopefully, can convince you, no, you do still get really good readout. Of course, with any kind of sequencing, you do have a slight dip, but it's just the enrichment and it's just the way short read sequencing does, but it still gives you the data you need without compromise. If you do have RNA that's much longer than, say, 9,000 nucleotides, then maybe you do need the long read sequencing method that combines with either our single cell methods or other methods. But in general, I don't think people would have RNA much longer than that. So this technique can be used in many, many research projects. And of course, like I mentioned, the cost is greatly reduced, and you don't need to combine two technology. You just buy one kit, and it's one single technology, one workflow. It just makes it easier. Okay, and the next product I wanted to focus on is the FocuSCOPE. And this product is very unique in the sense that it's very customizable. And what it offers is that you can enrich your region of interest, you can customize that, but you also get your whole transcriptome data on top of that. So if you have specific splicing isoform you want to look at, SNPs you want to look at, or a very rare transcript that that normally doesn't get captured when you do whole transcriptome sequencing, you can use this method to enrich that and then study them. And it allows you to capture up to 10 targets, region of interest. So you can design up to 10 that we can do, but we also offer pre-validated panels. So this is how it works. On our beads, you will have our standard poly-DTs to capture your mRNA, to give you the whole transcriptome. But then you can design up to 10 of your own gene of interest for enrichment. And we also offer pre-validated panels. So we have the blood cancer panel, the lung cancer panel, EBV, also an IB panel, and pleural hepatosis panel. As an example, because we do have many different panels, I thought I'll just showcase one. So with the EBV, as you can see here, if you're only just looking at your whole transcriptome, you do lose out on sort of EBV specific probe or specific genes. Whereas here you could see it gives you a much better differentiation and enrichment of the EBV. Okay, so finally, this is the last one I will talk about. So it's called MobiuSCOPE, and essentially it helps you look at glycosylation, which a lot of people find useful, especially in cancer research. It gives you not only the whole transcriptome, but also the cell surface protein glycosylation from the same cell. So you can be confident that they're coming from the same cell rather than doing two separate methods and then try to correlate them. So how does it work? It uses the covalent binding to LACNAC on the cell surface protein. As you can see in the beads here, you still get your whole transcriptome, but on top of it, you get your surface protein tags. With this method, you do your labeling before you do the cell partitioning part. So this is just a little extra step you do with this kit. So how does it perform? So what we've done here is comparing the three methods. So you got your control, which is the untag control. So that gives you the basic of all the different cells annotation here. And then we look at the sort of common technology that you would do. So you got your scRNA-seq and then our MobiuSCOPE. As you can see here on the right-hand side, you don't get compromisation of this sort of the composition, cell population composition. You don't compromise with the MobiuSCOPE, which you do see here. There's a huge variation with the scRNA-seq. So with the MobiuSCOPE, you can be assured that you will get a much cleaner biology, so the labeling itself will not disturb the cell population distribution. And you preserve your cell population much, much better than other technology. So as I mentioned, with all of our workflow is done on that scope chip, our core technology that I showed earlier, and it's a manual workflow. However, some of you might want to do many samples at once, or you want an automated system. We also offer that. So the idea is we want to make single cell accessible for everyone. It doesn't matter of your budget or your experience, but some people might still prefer automated version. So we offer that here. This is the Matrix Neo. You could do up to 4, and all the product that I've just showed you earlier can work on our Matrix Neo. And of course, it gives you more robust and consistent data because you sort of take away that human error part of the workflow. So hopefully you have found some of those 4 unique solutions interesting, and you might have thought, this might work in my research. But if not, I also wanted to showcase why Singaron is a partner you can trust. So as I mentioned before, we also offer services. So we are really a true single cell specialist. The learning from our service lab feed into our R&D, and we're constantly improving. And so far, we have sequenced over 1 billion cells and still counting. We have worked with over 2,000 sample types. So if you have anything that's very difficult samples, tissue types, we probably worked with them before. So do speak to us. We're always happy to talk to you about that. And we're also not species biased. So we have worked with over 70 species. And we have been entrusted with over 9,000 projects and over 4,000 customers in more than 20 countries. And we also have many customer publication and high impact publication, over 1,000 and still counting. So if you're interested in those, please visit our website. We upload them there, or get in touch with us. We're always happy to share some of these publications with you. And of course, finally, don't take my word for it. So one of our customers have used the MobiuSCOPE as one of our first customer to use it, from Georgie from Plurify, who's a co-founder and CEO. And it's a really cool startup company working on engineering long and complex RNA to help cell and gene therapy. And because of what they're working on, it's very unique. They have tried many different single cell technology. They've tried long read, and none of them were able to give them what they needed. So their RNA ranges in sizes, and it could be at different position along the length of your transcript, And so they tried out MobiuSCOPE, and it was the only one that can give them the data they needed. And now he has incorporated MobiuSCOPE as part of their QC and help them continue their research. And with that, thank you very much for listening to me, and I will pass it back to Irene.